and Pentti's going to be telling you about his model of sparse distributed memory and its relation to the cerebellum. And as I mentioned in the email, so the basic model that Pentti is going to be talking about is described in his book, which was published by MIT Press. And this is a really very elegant and mathematically well-grounded model of memory, of associative memory. It's related to architectures like the Hopfield that we discussed, but as you'll see, it sort of has a different way that is derived. But I just want to read you here a little excerpt that Douglas Hofstadter wrote from the book. So Douglas Hofstadter wrote the foreword to Pentti's book, and what he says actually captures very succinctly how I thought about it the first time I read about Pentti's work. So this is what Douglas Hofstadter says. This book, because of its pristine mathematical beauty and its great clarity, excited me deeply when I read it, and I believe it will have the same effect on many others, whether they are professionals in the field of cognitive science or not. Pentti Kanerva's memory model was a revelation for me. It was the very first piece of research I had ever run across that made me feel I could glimpse the distinct goal of understanding how the brain works as a whole. Okay, so it's not going to be the whole brain, it's going to be the cerebellum today.
Well, thank you, Bruno. And I'm afraid I might disappoint Doug Hofstadter with this talk and with any work that will be done for still a long time to come. But I would say it has been fun, it has been exciting, and I hope that you in your work will also be similarly excited whatever you decide to go and get into. There are a lot of interesting things to discover, and what I'm going to talk about right now is it was a discovery for me, but it turns out it has been discovered several times over, and described several times over. An interesting thing, the cerebellum, as I will describe it here, has been described in the same way, at least on two other occasions. So we have independently come to sort of the same conclusion. That doesn't necessarily mean that we are all right, but at least we are wrong. We have company if we are wrong, so which is nice. So I will. What I want to do is talk about one particular neural structure that, if you start thinking of it in information processing terms and computing terms, it really brings into mind our computer's random access memory. And so I will be using terminology and concepts from computers and mix them very freely about ideas, concepts, terms from neuroscience. I'm not a neuroscientist. I'm come to this more from computer science and engineering background, and the neuroscience part was sort of a late discovery in this work that I did, and it's just sort of added something unexpected when I noticed the correspondence. So first I will talk a little bit about the cerebellum, and then about the associative memory models that you have already discussed in this class, and then a little bit more in detail the sparse distributed memory, which is a specific associative memory, and then so how that maps into the cerebellum, and then on the other hand how it maps to the random access memory, and then I finish with some talk about the capacity of biological memory. If the cerebellum is interpreted as a random access memory, what is its capacity in very standard data processing terms, in terms of bytes? So see, we will go until what?
5.
Until 5. Okay. Here is sort of a sketch. In these pictures, there's often text. You don't need to try to read the text. I mean, it's just that they were in the pictures, and I haven't done math them. But just so anything that you'd have to squint or look hard at, just ignore. It's not meant to be part of this talk. But anyway, so here is sort of a sketch of the human brain and the cerebellum, and on the screen it is about the size of the brain. Of course, on that screen it is a little bit bigger, both the brain and the cerebellum, but relatively speaking. And this is how it sits: the brain stem, and there's the cerebellum. In terms of volume, you can see the cerebellum really, well, I don't know, maybe it's about 10-15% of the total volume of the brain. And here are some images of cerebellum. Cerebellum has been around already in amphibians and reptiles, birds, and then mammals. And in relative terms, it's fairly small there. Relatively speaking, it has been growing with evolution. Of course, with human evolution also, the cerebrum has been growing. So in terms of, relatively speaking, they're probably about the same in those, I mean, the relation of cerebellum and cerebrum. So anyway, it seems to be sort of hidden back there, and in many neuroscience things, it also gets to be hidden. But I think it's a very interesting structure, and that's I'm trying to explain in which way it is interesting. Here is shown, if you look at the previous picture, it's very highly folded. Well, of course, this is always folded, but this is even more highly folded. And there are some cross sections showing how in different animals these all are, well, the birds and mammals. Anyway, and all of them is very, very highly folded, convoluted, which means that in terms, I mean, there's a certain volume, that volume packs a lot of surface area. And it's the surface area, or the surface that is the cortex that has the circuits that I am talking about. Here's one of the folds, sort of schematic picture of it, how it is structured. The most prominent cell type is the Purkinje cell, and probably is most prominent because it probably early on it took the stain very well, and so it became very visible when Cajal was sketching. This goes back, over 100. When Cajal, 100 years ago. So he mapped this circuit very, very well, and so the main cell type, the Purkinje cell, here shows one of them, here in another. So they just, if this is the surface, and then these are also, but this is side view and side view. One is facing. I'd say these are facing, and then this is side view. So they are very flat. You could think of it like your hand, and then they go like this. We'll see more. The other very important cell. Okay, first of all, this is the only output from the cerebellum. So Purkinje cell. Here is one output. Here is one output. Each of those produces an output. There are several kinds of input, and the input come from other parts of the brain, and they come along what are called as climbing fibers. Massive fibers. And it's a massive fiber, and then it connects in the cerebellar cortex, connects with these granule cells, and these they are like little critters, so insects, and there are huge numbers, and we'll see in a slide a few lights forward. The other important input I'll be talking about is the climbing fiber, and it's very interesting. It's topological very interesting. You have a Purkinje cell that produces an output. You have a climbing fiber, each that just goes along this, and then it forms synapses all in that arbor of the Purkinje cell. So each Purkinje cell has its own climbing fiber, and this pairing of output and input, it's topologic. It's very important in trying to understand or speculate what the circuit might do. Okay, so what happens is that mossy fiber comes to the granule cell. The granule cell, then it goes in this layer, and it intersects a very, very large number, several hundred. Each of these intersects several of the Purkinje cells. In this area, they call parallel fibers, because they like if you go see some telephone pole, and it has then they are like the wires that go from one telephone pole to another. So this is like the telephone pole, and the parallel fibers are there. Then there are other cells that I will not talk much about, and it seems like their function very much is to keep a general level of activation within certain limits. If there are not enough active neurons at one time, apparently it means that there's not enough information to sort of pull out, get the reasonable output. So these basket cells and stellate cells, they regulate. It seems like they are in a position, and their nature, inhibitive nature, is such that they could very easily seen regulating the amount of activity of the in the end, the amount of activity that Purkinje cells, how many Purkinje cells would fire at any given time.
So the stellate cells and the basket cells are inhibitory?
They both inhibitory.
And the climbing fibers?
Climbing fiber is excitatory.
These are excitatory.
Yeah. Climbing fiber, if a climbing fiber fires, it produces an instant firing of the of its Purkinje cells. So it is sort of like bam here, and then the response. It's just one might wonder what good is that. Well, I think it anyway. It looks like it is appropriate for heavy and learning. Heavy and learning, meaning that in the it sort of teaches these synapses in the presence of certain parallel fibers being active. It teaches those synapses to change their weights, and it is caused by now. And it could also be thought that this is if the Purkinje cell was firing incorrectly, the climbing fiber could be like a correction signal. Well, you are supposed to fire now, and if that courses, then course the corresponding synapses synapses at that time change their strength. Here is sort of the same circuit, only the other cells sort of have been pretty much weeded out. So you can see that the Purkinje cells, they don't necessarily in these kind of nice rows, but anyway, they definitely lie on these flat and perpendicular to the parallel fibers, and the parallel fibers get their input from the mossy fibers. These inputs come from other parts of the brain. And so you could think of it as, okay, here signal coming in, it gets distributed. Some of these parallel fibers will fire, and that causes some of these positive synapses in the Purkinje cells to start summing up, and if there's enough total activation, the Purkinje cell will fire in return. I like this picture because it sort of shows not just a single neuron or single Purkinje, it just shows how they are stacked. It also gives a pretty good idea of the orientation. And here is some detail. This could be the Purkinje cell dendrite, And here, parallel fiber, they just go here, and these would be the synapses. Of course, then the parallel fibers will also give input to other cell types, and that could serve as a regulatory mechanism for having a certain level of activity overall coming from the cerebellar cortex. Here's sort of a further sketch of the same, but I will use this later in later pictures. Here's your soul output, the Purkinje cell, and it's climbing fiber, so it goes all over that dendritic tree, and here are the inputs coming from outside the cerebellum, and then they go with granule cells and go into the parallel fibers. I want you to feel free. I think there'll be plenty of time for discussion. So if you want to stop and sort of ask, and if I can't answer it, maybe somebody else can answer it. I can probably answer something that has to do with the anatomy and architecture and things that have to do with the physiology. I'm not very up to date on.
It sounds like overall the inputs to the system then are the climbing fibers and the mossy fibers.
Those are the inputs that a computer scientist would look at.
Okay.
Exactly, yes. A computer engineer, rather.
As opposed to, what other? There's those other.
These are other inputs, and exactly what their function is and how one interpret them, I really don't know, and
so then we have these inputs, and then the output, as you said, is the Purkinje cell.
It's Purkinje cell. That's the output, and it goes first in a deep part of the cerebellum, and from there into.
And does it affect there in the deep part of the cerebellum? Is that excitatory synapse? Do you know, or inhibitory?
This one is inhibitory.
That's inhibitory.
Yes.
So Purkinje cells inhibit whatever they contact.
Yeah, that's inhibit. It's just whether it is 0 or minus or plus or 0 or 1. It doesn't matter because it can always be inverted to be one or the other. Whatever is needed down the line. And so, in this kind of circuit diagram, like in computers, if you turn zeros into ones and ones into zeros, you still have an equivalent computer. Well, anyway, this is something I've often felt, and people say, okay, inhibitory things somehow they prevent things from happening. That's not true, because they allow. When you inhibit certain things, you allow other things. So inhibition doesn't mean preventing. It's just preventing only in very local sense, not preventing in global sense. So if and I've seen in okay, these are inhibitory cells, so they therefore at some macro level something cannot happen. Well, sometimes that's inhibition allows some things to happen at macro level. So something at the micro scale doesn't mean the same at the macro scale.
So are most of the other inputs inhibitory, or are there other excitatory inputs?
The Golgi cells, they are inhibitory. The stellate cells, no, this is a basket cell, that's inhibitory. And the stellate cell, that's inhibitory.
Okay. And where do the inputs come from? The climbing fiber, the mossy fiber?
The mossy fiber inputs come from other parts of the brain. The climbing fiber comes from inferior olive, which is part of the cerebellum. And then where does that get from? I don't know the anatomy beyond that. And it doesn't mean, I don't mean it is not known, I just don't know the answers. And I think, the names that are associated with these models of the cerebellum, Albus and David Marr and James Albus, and I think in David Marr's papers, some of these others are also discussed. I may have read them, but I certainly have forgotten what the details are. Then down the line from here. Okay, here is just one sort of picture of a Purkinje cell, just showing how, when you look at this way, it just proliferates in a very, very broad arbor, and then the parallel fibers, and they call perpendicular. And the next slide shows, here's a dendrite, here's another dendrite, and all these little nooks then would be potential synapses with the parallel. The parallel fibers, all this white stuff, it's full of parallel fibers. It's just packed with parallel fibers. It is a very dense three-dimensional structure. It's like a memory stack. Now here you can see there's a lot of small text in there, so you don't need to read this. But anyway, this is one way, and engineers have started looking at it. You just what different cell types they are, where do they get their inputs, where do they provide their outputs, in what numbers, what's the number of this any particular cell in the cerebellum, and there are lots of question marks. In other words, some statistics are known quite well, and other statistics they just at least were not known very well at the time when this was assembled about 20 years ago. But anyway, this was done by an engineer, Egon Lebner and David, and engineer likes to see connections and numbers. So this is an engineer's view. Now here are some of the numbers we would like to learn to appreciate of the various kinds of cells in a human cerebellum. The granule cells, the ones that receive mossy fibers and then go up into the outer part of the cortex and branch into parallel fibers. There the estimates are anywhere from 10 to 100 billion, and it has been jokingly said that of the 50 billion neurons in the human brain, 100 billion are granule cells of the cerebellum. So anyway, they are very, very numerous. Of course, they're very small, so a certain volume can pack quite a lot of them. Whereas the Purkinje cell is very large, and so they are only 15 million. Well, only 15 million is a large number if you actually start counting them, but in contrast. And then these other cells that are thought to sort of regulate the level of activity, they too run sort of in the 15, from 1 million to in that range. But the interesting numbers to me would be sort of these two, the 100 billion and 15 million. Yeah, it's just, in other words, yes?
So the granule cells, their role is basically to connect the Purkinje cells to each other, and the Purkinje cells are the ones that are performing the actual computations.
That is that is true. The Purkinje cell, I wouldn't say that the granule cells connect the Purkinje cells to each other. I wouldn't say that. The granule cell or its parallel fiber, it intersects a large number of Purkinje cell dendrites, but no information flows back and forth from one Purkinje cell to another through these fibers.
Oh, okay.
So they are parallel fibers are connected to many Purkinje cells. So topologically it connects them, but in terms of passing signals, no, they are just the Purkinje cells that receive the signal from the parallel fiber.
Parallel fibers are kind of like a bus.
You could think, yes, yeah.
What's remarkable about that number is that, this number of granule cells, right? Even if they're just 10 billion, right?
Yeah.
There's not only that many cells, but each of those granule cells sprouts a parallel fiber.
Yeah, I think
that means there's that many parallel fibers that have to kind of all.
What would be the best?
there to-
What would be the best picture to show
Purkinje
this?
cell dendrites.
I guess this is the best
So for
thing.
every one of those granule cells, there's a parallel fiber.
Yeah, once you come from a granule cell, and this goes, each of these parallel fibers intersects about 500 or so
500.
of Purkinje cells. Yes, and of course you have a sheet. I mean, if you spread out the cortex, it becomes a sheet, and no single parallel fiber goes through the sheet. It just covers a certain area or certain length, and then they just locate it throughout the sheet. But these parallel fibers definitely give a direction to the where the folds are. So the parallel fibers run this way, and then the folding is sort of happens parallel. So that's. Okay, now that we have a little bit of a review of what the cerebellum looks like in terms of its connections and in terms of numbers, we'll go back to things that you have already discussed in this class. And you discussed associative memories, and here is one of the very early pictures that appeared in the literature on associative memories. And in this case, what the associative memory was trained to do, it was trained on these complete images. And once it was trained, then it was given a noisy image. In this case, the noise was just randomly changing some of the pixels in the original image. In this case, masking off some of the pixels in the original image. And then when this noisy image was given as input to the associative memory, it would produce the original image. I mean, after having been trained with the noise-free image, a noisy image would retrieve the noisy image would be a noise-free image. And same here. And it wasn't. I mean, the particular memory at the same time did hold this phase and it hold that phase and several other phases. It was not just that it was trained with one phase. No, it had several of these images at once, and so with the noisy version of the same image, it could retrieve the noise-free version. And that this is the diagram structure of the network that produced these things. You would have a raster of 54 times 56 pixels, and in training you would put the same pattern in these two I call them registers, vectors, and then produce the cross product of the pattern with itself, and you add it into the matrix that stores the associative memory. And after having stored several of these images in this fashion, each image you just string it up into this long vector, and then produce with its cross product with itself, and you accumulate a matrix that holds these these images. And then if you want to come with the noisy image, would be the noisy input pattern. You put it in here, and then you do a vector times matrix multiplication operation. You get a vector of sums, and by thresholding or a squashing function, in this case it's not a thresholding because the various values are not binary. If you look at the picture, the pixels are not binary. They are a lot fatter and smaller, and so here it shows that you get various levels of darkness, and then of course in this long vector you just do it like a TV scan. You just distribute it over that picture, and then you get the the resulting picture in the examples, a noise-free picture. These kinds of memories, they were some of the exciting things that were studied at the second wave of artificial neural net research, and people like Kohonen and this is James Anderson, Brown University, Will Shaw in Scotland, what is that university? Edinburgh. Hopfield. These names, I believe these names you've heard these names in your previous lectures here. The very first memories, they were just linear, and you just and in that case you just don't get different bases of attraction. But when you go from the sum vector sum or the vector matrix product through a squashing function, some nonlinear function, then you can store several different patterns into this kind of memory. So this is sort of just like a review of what you have discussed here. Now I will go into networks of slightly larger and more interesting, in the sense that now you have that particular had one set of one matrix, and it would be the matrix of trainable weights. Here is how now quiz. Is this a two-layer network, or is this a three-layer network? Well, if you go back into literature, some people call this a three-layer network, and some people call this a two-layer network. I don't know. It's the interesting thing is that it is described by two matrices. Here is one matrix. I mean, these are like input units, hidden units, and output units. It turns out you really don't have three sets of, in these models, you don't have three sets of neurons. You have just inputs. But then you do have hidden units and you have output units. So I still call this a two-layer network, even if you'd say that these would be the layers and these are the connections between the layers. So anyway, that's it. So this is how they're often drawn, but I don't think this is a very helpful way of drawing the picture and thinking about it. So I've taken the things here and sort of start morphing them. And here would be like the input units, and then their weights, and go into the hidden unit. And I first morph it instead of having the weight sort of sitting on those connections, I just put them into a vector, and so inputs, and then there's some sum operation, and then a nonlinear threshold function, or some some nonlinear function that then produces the output. And so when you draw them this way, I mean, this is just the same thing. And well, that's not exactly. I've put this, taken this part from here and put it down here. And then this one, just flip it over. Well, flip it over in 3 space. And now what we have is we have something that you can start stacking next to each other. This concept is these pictures. And then with these, when this thing is drawn like that, then you can stack them. You know, if you look at here, these is stacked this way and they're stacked that way. I mean, the one on the left is a stack of those, and the other on the right stack of those. So this is a nicer way of drawing the same picture, and you could think of these as, I mean, each one of these is a neuron. This is this dendrite, and there's a cell body, and then the axon going out, and there's a dendrite and cell body that does its magic, and then axon going out, and the synapses then would be in those little boxes, in those cross sections. So this begins to look like vectors and matrices, which I think is much nicer because that's how we like to do the math and think about them. So I've just put them side by side, these two ways of drawing the same picture. And so from now on, I will really use this as form and give it an interpretation. Okay, here is a neural network with one set of hidden units and one set of output units, and here is sort of the same done as more like a matrix, and it's also organized and labeled a little bit like I could label a computer memory, computer memory. There's an address register, and there are some data registers. There's a data in register and data out, or word out. Computer word going in, computer word going out. And the way it works when you want to store information. Okay, yeah. First of all, in this sparse distributed memory model, these two matrices are very different, have a very different function. The left matrix is random and it is fixed. Once you build it, it will not be changed from then on. And then the right matrix, it is a matrix of variable weight. So when you train something, you are training the weights of this matrix. So what does this do here? Why do we have here? Well, the thing is that here you have, I'm thinking of like 1,000 bit inputs. What this allows you to do is expand this vector into an activation vector that has a much higher dimensionality than the input. So here, thinking of this as being 1,000 bits, it has been expanded into million bits. And so when you put something here, it will, this vector, input vector, will produce an activation vector that is very high dimensional, but also very sparse. And the sparseness turns out it is mathematically it's important that this intermediate vector is very sparse. And then the ones in here indicate the locations in the contents matrix that will be activated and affected when something is written into the memory, or when something is stored into the memory, or when something is retrieved from the memory. So with this preamble, so this is what happens. You want to write a certain pattern, bit pattern. I'm thinking here just plus and minus being like zeros and ones, ones and zeros. So you want to write a certain pattern, you want to store a certain pattern, and you want to use certain other pattern as its retrieval cue, or its. And so what you do is you put this retrieval cue, you treat it as a computer memory address, and it is compared to all the rows of this left matrix. And compared in terms of Hamming distance. Hamming distance means where the number of places at which they differ. I mean, if you start computing the Hamming distance between these first, okay, this you get 1 from there, 2, nothing, 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 nothing. So of the visible bits here, the Hamming distance between those two would be 2, but because they're invisible bits, the Hamming distance is 501. Here it is 440, 5. And then the ones where it is the low, where the Hamming distance is the smallest. In other words, of this set of random vectors, it selects the ones closest to that pattern and activates. I mean, here it's shown here, those 3 as having been activated. Okay, then so that's how you get started. Okay, those are the locations, those are the places in memory where something will be stored. And what is being stored is coming from this other bit vector here. And whenever there's a minus, you subtract. Whenever there's a plus, you add. But since this vector has previously had some other content, after adding this one, you get some. But if you look at this one here and this one here, apparently this particular vector, before this storage operation, it was all zeros. But when you subtract 1 for the minus and add 1 for the plus, then after the write operation, the storage operation, this particular location got this content. And so that's how you go through your data set. You have a certain pattern that you want to use as the retrieval cue, and then the pattern that you want to retrieve at cue. You keep adding into this matrix, adding meaning, adding and subtracting. And then later on you want to come and retrieve, and you can use here again with some pattern as a retrieval pattern. So it again will activate some locations. If you use a previously used pattern where previously something had been stored, it activates the same locations, and then from those activated locations produces a sum, column-wise sum, and after threshold, then you get a binary. In the end, you get a binary output. So that's how this simple memory works, and it works an awful lot like you have had, I don't know, Hopfield-net or Wilshire-net, but the only difference is that you have this intermediate matrix which starts, takes certain length vector and just blows it really up, and what that does is it makes it possible to store a very large number of vectors in the right matrix. So that just shows the comparison of the pictures. It's just the same picture done. One done with symbols, another done with real data. Okay, and this, if you look at sparse distributed memory, that particular memory, associative memory from the outside, so it has a certain number of input units. It's the address register, output units has the word out register, desired output. It goes in from the data in word in register. Inside there are hidden units, and they produce the activation vector, and each hidden unit is called hard location because you actually have some physical stuff that represent that particular address of memory. So these are the concepts that that are used in describing the memory. Row output, you get the sums, and from the sums by some thresholding function, you get the final output. And the operations, okay, this is what is inside, outside, and then if you look at it operationally, you have the operation of addressing the memory, you have storing into the memory, and retrieval from memory. That's sort of very computerist talk. Okay, so now here, I put this here just to show you the difference between this kind of two-layer associative memory and then the one that produced the very first picture. And as you see, the size here, it's when you use the same vector in the input as sort of desired in output, and then as the Q, then the number of adjustable weights, it is limited by the size of the vector, by dimensionality of the vector. Here, when you have the hidden layer, the amount of information, the amount of number of synapses that are adjustable, it becomes independent of the dimensionality of your data.
So even if you were using it in auto-associative mode, so the word in register, let's say, with the same pattern coming in the address register.
You could put the face here, and you could put the face here, and train it that way, and you would produce those same pictures. You would be able to produce, and we have produced the same kind of thing with this.
Have much higher capacity, perhaps.
But the thing is that the capacity is not limited by how large your picture is. Here's a little bit of sort of how, in sort of abstract mathematical terms, how it works. If each one, okay, first of all, if you have like 1,000 bits of address, in essence you have 2 to the 1,000 possible addresses, binary numbers. Now, 2 to the 1,000, you'll never build a memory that has 2 to the 1,000 locations, because there just isn't enough stuff in the universe to build, or much less in the brain, to build that many locations. So by choosing randomly, just putting these just sort of randomly, sums, okay, this represents, a large circle represents the total space of 2 to the 1,000. And only some of those addresses in that space actually will be represented by a physical memory location, by a neuron. And each row in here is one of these boxes, and so it shows that not nearly all, I mean, infinitesimal part, fraction of the possible addresses will actually be represented by a real bit of physical memory. And then when you give this memory a retrieval queue and address, there's almost certainly not going to be a location, actually hard location, piece of hardware with that exact address. So what you do is you sort of activate all the memory addresses and locations that are within a certain distance. It is this Hamming distance. And so that's what, I mean, these things that fall within a circle sort of correspond to the 3 rows that are activated.
So they're within the Hamming distance, then the corresponding element in Y becomes 1.
Yes.
And so then what happens to the contents matrix?
Okay, if it is a storage operation, you change the contents, that particular row, each of those rows you change according to what is being stored. You add and subtract. If it is a retrieval operation, you sum the activated rows, and that's the sum here. And then you do a threshold. It may actually, some of this becomes even clearer when I draw this next to an ordinary computer memory. That picture comes. But did you have another question?
No, I guess that's it. I still kind of don't get it.
I think having a comparison to a RAM.
Yeah, I think we with the layout, we have laid out the groundwork.
Yeah.
Okay, now, but before that, okay, here, this shows, if we're writing into certain location, that particular address will not have any hardware associated or any physical stuff. But so we take certain Hamming radius, and within that radius we put data into each and add data into each of those. Then when we read it's at this time with a different address, different queue, then okay, we take again all the locations that have addresses within that same particular Hamming radius from that one, and then you add. So writing means that you increment and decrement the contents of those locations. Reading means that you sum up from the activated locations and then do a threshold function as the result. So this this shows how if two addresses, the writing address and the reading address, if they are very close to each other, the intersection is large. If they are very far from each other, the intersection is very small. And that's how, when you have a large intersection, you can recreate an old pattern. When you have a small intersection, then it just all looks like noise, and so you are not recreating a pattern that has been stored. And as you should not. I mean, if you are not reading with an address that is similar to what you have written into, you should be getting noise. Yes?
So is the address matrix just random?
It's random in this model. It's in the very base. That's yes.
Okay, and it's fixed in stone.
And it is fixed. Once it is fixed, when you come, if you later on come, 10 years later come with the same address, it would still activate the locations that previously got that data. And it turns out if you don't have it fixed, you're sort of building on sand. And I think that is actually, it's an important feature of this architecture, and we'll see later that this is actually how the cerebellum also seems to do it. So now, in the next few slides, I'm going to map this sparse distributed memory that we have had here under the cerebellum, and for that I've drawn a picture with sort of same thing in three different ways. I mean, here you've already seen this. I mean, this would be like the dendrites in a cell body and output. So and here are the inputs. I call this, because this looks like a a column in the matrix. I call this the mathematician's column. Now here is shown the same thing that engineer would draw. Okay, there's some wire coming in, and there are other wires going in this way, and then there are some locations, some latches, some flip-flops that are affected by what's coming through these various wires, and then there's a wire coming out, and so here is your one input wire, output wire, and here are what call it sense wires. So this is the engineer's column, and here's the biologist's column. This is being the cell body, and this is being standing for the dendrite, and this standing for the axon of the climbing fiber. So here you can probably tell what are you an engineer or mathematician or are you a biologist by how you prefer to look at this picture. So I've put the Purkinje cell sort of in the same fashion as the as the biologist column before. So the climbing fiber, and then it's an axon, and it goes all over. It just climbs all over the dendrites of the Purkinje cell, and then these are the sensing wires and these squares stand for synapses that where the parallel fiber actually meets and makes a synapse with the dendrite of the Purkinje cell. So to make that particular picture match what we have seen before, previously I had the same part here sitting there next to it. I just sort of cut it and put it that way. The reason is that, okay, first of all, here is your mathematicians' columns, and here are your biologists' columns. So I want you to imagine that these things here, the Purkinje cells, each one of them corresponds to one of these columns. Each Purkinje cell, here there are only one of them, and here several are shown. And the climbing fiber coming in, and then here with the granule cells, they get their mossy fiber input, and they have those little very special little critter kind of structures, and then they become the parallel fibers that go into the Purkinje cell. So these are the parallel fibers, granule cells. So granule cells, parallel fibers. It's a and now, if we look at how successful this mapping is, and there's a major agreement in things like the input and output dimensions. The input and output dimension means how many mossy fibers come in, how many climbing fibers come in, and how many Purkinje cell output come out. So the dimensions in the memory model they were just 1,000, but in they could be million, but they are of that order. And another very important thing is that there's a one-to-one matching between read and write data lines. So here is when you're writing data, and here's when you're reading data, and so it's just paired one-to-one, and they are paired one-to-one, and that is to me that's a very strong telltale sign that this is a programmable memory location, a programmable synapse, and it is programmed by simultaneous action of the climbing fiber and the parallel fibers.
The W's are the things you're writing into the memory, and that's the climbing fiber, right?
Yes.
Okay.
Yes.
That's what you write in, and that's the climbing fiber coming in.
That's coming in.
So the idea is that goes up the dendrite, and then there would hopefully be one of those sitting in every conjunction between a parallel fiber and a previous cell dendrite. Then there'd be a climbing fiber there that would potentiate that synapse.
Yes.
Okay.
Yes.
Raise the value or decrement the value.
It change. Yeah. Right.
Accordingly.
Yeah.
Okay.
Well, you don't need the actual climbing fiber to coincide with the actual synapse, I think. It's just that it fires the whole thing.
What I've read about the climbing fiber action, it just produces a very predictable and strong response firing of the Purkinje cell. But I guess by going all over the place, it really catches the whole arbor. So wherever they happen to be programmable or adjustable synapses, that part of the dendritic tree has been activated. I think that's the. Yeah.
But it seems like what you want. So, like, if a parallel fiber is active, then it's going to increment or decrement the counter.
In the presence of input from.
Yeah, and if the parallel fiber is not active, right? The vast majority are not active.
And
then for all of those, there should be
nothing. Yeah.
Synapse, irrespective of what the climbing fiber is.
Irrespective of what the climbing fiber is.
So it's the parallel fiber that's gating the plasticity then.
Yes. Mm. And also, see, Purkinje cell can fire under two conditions. One is simply on the basis of the parallel fiber input, whatever comes from there, and that's your normal retrieval operation. And that firing is different, physiologically it's of different nature. It is not nearly as strong as the one when the climbing fiber gives it input. It really puts that cell into shock. And maybe it's enough of a difference that in that particular shock state, it is high enough for the synapses to potentiate. Whereas if it fires on basis of simply the parallel fiber input, it is much milder, and apparently it would mean that the synapses don't change. Actually, this maps even to this memristors in a different way, that then you can get the information out, but you are not affecting the value. So that pairing, from an engineer, that's a real clear telltale sign that those two would be paired together, because then you can actually train. You can train. You can change the data according to what you want to read, want to see next. And then another telltale sign is this huge number of these hidden units or these granule cells, which means that this would give it a very large capacity, and independent of the size, the input vectors and output vectors that come from outside the cerebellum. And another one is the parallel operation. So these all are sort of very confirming the mapping that I have described. There is also a major disagreement, and that is that in the cerebellum itself, if you look at it, we'll still a little bit later see a picture. Each of the granule cells has only 3 to 7 inputs, and not as in my picture it would have 500 to 1,000 inputs. So only 3 to 7 inputs, and then also at the physiology that input lines coming that they are only very sparsely active, 1%, 10%, 1% even may be high. I mean, the amount of massive fiber activity that comes into it is probably under 1% at any one time. So, and whereas in the model that I've shown you, it's 50-50. You know, half of them are active, half of them are inactive. So there's this kind of disagreement with the model, but it has been mathematically shown that actually that all is overcome, and this hyperplane design assumes that only a very small, in the inputs coming here, only a very small number are active or plus, and mostly are inactive at any one time, any one pattern. So the patterns are very, very sparse. And the other thing is that this random matrix that originally I showed as being 50-50, zeros and ones, actually can have only very small number of connections. Here I'm showing that each line would have two or, well, you can count to ones in that area. It would have a very small number. And this turns out to be mathematically actually better design than the original design I showed in previous slides. So there was this first agreement from simple sort of wiring and neuroanatomy, and then mathematically shown that what actually nature has come up with, in other words, granule cells that have only a very small number of inputs, turns out to be mathematically a much more efficient way. And of course, in terms of energy consumption, is much more efficient. So now we'll see how the sparse distributed memory corresponds to random access memory. This is one way of looking at the RAM. Everyone has a RAM in their computer and cell phone and whatever. So here you have memory. You can think of it in this way. You have a memory address, and then you have, for each possible address, you have one location. So here it's a location 0, 1, 2, 3, 4, 5. And if you have 20 bits of address, then you would have about 1 million. The last address you reach, you'd be 1 million. And so you do have one memory location for each one of those. So 20 bits would require you to have 20 bits of address would require you to have 2 to the 20 memory locations. Then when you put in an address, it activates only one location. This is because now you have every one of a possible 20-bit address actually is represented in this matrix. It means that it can select, once you give an address, it selects that particular location. Whatever data you want to write in that, it goes into that location, nowhere else. It destroys whatever was there before. Then you put an address later on the read, it activates the location and copies the data. So that's one way of looking at random access memory. This in your RAM, this actually is present there. This is the left part. It is implemented as sort of a decoder circuit. Its effect is that you give it one address, it selects one location. So you can think of it in those terms. So now I've just drawn those two next to each other, and I don't know if I need to really say much more. So, okay, I guess that's the interesting thing to notice. So any single address activates one location. When you put here 20 bits, you put 1,000 bits here, it activates several locations. And then what happens in terms of reading and writing, we discussed. The activated locations are either modified or they contribute to the output. Just like here, the activated location is modified when you're storing, or the activated location contributes to the output. So that's how how similar the random access memory and the sparse distributed memory are to each other. And so I once more draw this in this particular fashion to show how those, the cerebellum and the sparse distributed memory sort of can be mapped to one another. Okay, let's see. And I think this is just a nice last picture of this kind to finish with. It just shows how then they all this is sort of laid out. And again, I want to point out this very small number of inputs. There's one more feature which I have not mentioned and should mention. These synapses are not modifiable. In fact, they are very. They first of all, there's a very small number of them, and okay, this is excitatory. Okay, so so it gets a small number of inputs. Let's say if it has 3 input synapses, then this particular granule cell probably fires if all 3 of those synapses get input and no other instance.
It's known biologically that those are non-modifiable. I mean, that's actually shown in physiology.
There is. I don't think anything is known positive, but the way they are physiologically structured, this shows because there's one more axon coming. I think that axon comes from the Golgi cells, and it is a very special kind of synaptic structure. There isn't much to modify there when there are only 2, 3, or 4 things to start with. But okay, well, maybe somebody will come up with the definitive answer to that. Well, 20 years ago, some neurophysiologists or 25 would swear that these synapses are not modifiable. This is second or third hand knowledge, but there's a conference of neurophysiologists, and one very famous one would stand up and swear there's no modification of synapses of the Purkinje cell. Now, this kind of model would say if anywhere there is some reason to be modifiable, it would be right here. And I think later research has confirmed many times over that these synapses, they disappear or grow. I mean, so people's opinions change over time by what they find out. These were not supposed to be modifiable. Now, I mean, by the mathematical model they should be, and I think physiologically, by mathematical model these should not be modifiable. And it means that things will not change. I mean, the world you learn when you are 5 years old, it still has the same meaning. If certain receptor gives you certain input pain or something like that, still pain 50 years from now. That's how I look at this model, saying that something has to be built in and stay that way so that everything doesn't change. Its interpretations of a long time can remain the same. Here's a few more things about the capacity. So the capacity is then related to the number of synapses between the Purkinje cell and the parallel fibers. And because there's such a huge number of parallel fibers, and the Purkinje cells, the arbor is very large. There's a very large number of synapses, and we could say that if we allow memory to produce a small number of errors on retrieval and train it without a product learning rule, then the capacity is it's about 10 to 15% of the total number of granule cells. The granule cell is the one that marks a memory location. It's the one that activated and then runs through. So then it turns out that if you do some mathematical analysis, you can actually store much more. But well, this means that you have some sort of error correction, and since the synapses are not binary, then you can actually store. If you're storing binary patterns and your synapses are not binary, then the number of patterns stored can actually be even 100% of the number of memory locations, because each memory location, each synapse can have several values. If your synapses are binary, then you can never get to 100. Certainly you can never get to 200%. I mean, that's... Okay, so now I put sort of biological memory and computer memory sort of contrasted with each other. So here's your computer memory. I put 28 bits of address and 32 bits of data, and so you have 1 gigabyte, and cerebellum memory would, well, these are some numbers corresponding to some of the you might think that the inputs would be like 7,000 bits, and the data. Anyway, the cerebellum would have capacity for 100 gigabytes, and that sort of gives you an idea of the scale of biological computing. And so here's your 1 gigabyte memory. It's just like how much information in one book. And no, that's 1 megabyte. 1 gigabyte. So you have a 63 shelf feet of books there. And the cerebellum seems to be something like, if it is like 100 gigabytes, it's like 100,000 books, you go 1 mile. So if you can store 1 book's worth of information in your memory one day, you have several hundred years of worth of storage. I mean, that's the scale of biological memory, and I think our modeling, really, we should sort of see, are we modeling things, thinking of things in those terms, in these huge amount of data that could possibly be stored. And so how do you organize it? How do you access it? What do you... That, to me, is very big part of the challenge of computational neuroscience. And here are some references if you want to read some of the work that, where some of these things, where each person first sort of discover it and how they tell their story. So, thank you. This is. There's much, if you edit 5 days, and much time for discussion.
Well, you just hang around a bit and people...
I will have to be in the bar at 5:30.
Yeah,
so.
me too, absolutely.
Yeah.
We'll hang around for a bit.
And if any of this kind of stuff is interesting to you, I come to the Redwood Center, regularly, once a week. I've been coming, and I'd be very glad to talk. And if you're interested in modeling of this kind, I'm open to discussion.
See you all on Monday. 2 o'clock?
Yeah.
Okay. Great. See you then.